First, I want to thank the organizers, obviously, for having the opportunity to be here and sharing our technology from a biotech perspective. It's actually very, very humbling to be in the middle of brilliant scientists that are showing a lot of great new things to try to tackle cancer specifically and beyond. My name is Raphaël Ognar. I'm a CEO and co-founder of NKILT Therapeutics. And I will go through the technology that we have created with my co-founders, fully owned, not a spin-off from university, but that should allow us to really tackle more cancer patients across multiple types of cancers by developing a novel off-the-shelf cell therapy. Nobody needs to be explained how cancer can be debilitating and impactful nowadays still. But more importantly, despite the incredible progress we made with cell therapy particularly, and specifically CAR T cells, we still face humongous challenges, essentially, of course, from a potential resistance mechanism, but I think more importantly, by the fact that the current autologous CAR T technologies are clearly not scalable and not going to be able to roll out to larger patient population. And a striking statistic across the United States is actually making us focusing on how we can bring more accessible therapies to patients, because only less than a third of patients eligible for CAR T today are receiving a therapy. Most of them, either they cannot access, they cannot afford the cost, or even worse, they may lose eligibility or die throughout the manufacturing process. Because of this, with my co-founders, we decided to create an off-the-shelf chimeric ILT receptor, which is a CAR-like, to simplify, to engineer natural killer cells by the targeting of HLA-G. And that's very specific to the target, to be sure that we bring more accessible options to patients. The interest of HLA-G is that it's really known as a potent immunosuppressant. I'll come in a minute towards this, but it is expressed in over half of cancers. And you can find it almost everywhere in most dominant form of cancers. Our unique proprietary technology called KIR is based on a potential risk of tumor escape that HLA-G is involved on, and is actually going around this. HLA-G is expressed in 7 different isoforms, which give them an opportunity, give the cancer cell an opportunity to do escape mechanism by expressing different isoforms, which will make, for example, an antibody irrelevant at some point. The use of our specific engineering and also patented unique engineering to enhance the killing and persistence of natural killer cells is making a huge impact, and I'll share some of our data on this. And lastly, the importance of off-the-shelf to ensure that every patient around the world could have access to cell therapy. NKILT is not just me, obviously. It's a team of dedicated people and experts. Besides my 2 co-founders, I have a diverse leadership team that is joining me to try to move the needle and accelerate the pace. But I want to always use this slide to emphasize my 2 scientists that are in the lab since the beginning, since we started a little bit more than 2 years ago. And that are really not only incredible scientists, but they are also extremely dedicated and extremely talented. And hopefully, when we're going to close some funding, we'll be able to rapidly hire more to expand the team. But also on my side, I have incredible leaders that are seasoned in the world of cell therapy. On our board, we have a clinician, Dr. Barton, that has a significant expertise and experience in designing early first-in-human clinical trials in cell therapy, and also Carlos Martin, that is a business expert, currently chief commercial of Rocket Pharma, which is a gene therapy company. We will bring a new board member with our seed round when we complete it. And we have the privilege of having Sargil and Max Mamonking, 2 brilliant immunologists, and Sargil also MD, PhD, that keeps seeing patients on a weekly basis, hematologist from UPenn. He was, of course, by the side of Carl June for the first CAR T. And he has co-founded 2 cell therapy biotech also. So he knows beyond academia how to also move the needle to help us. So 2 words about HLA-G. It's a potent immunosuppressant, as I said. It's actually known to be the pregnancy molecule. It's what the fetus and the placenta are producing to shield themselves from the immune system of the mother. So we are all the proof that it works pretty well, and it works pretty steadily because it allows the fetus to grow without being attacked by the immune system for 9 months. Cancer cells have figured that out. And over half of them are using this exact same mechanism to shut down the immune system, making the cancer cells completely invisible to all arms of the immune system. As I highlighted earlier, the 7 different isoforms can make it difficult to target. Actually, the data on HLA-G is pretty scarce and I would not say inaccurate, but not great, because likely of not having good reagent to be able to target and identify the expression of HLA-G across all the different isoforms. But HLA-G 1, 2, and 5 are the most dominant one, and you can see that they're significantly different from one another. So if you use an antibody that will be very specific, it will work until you have another HLA-G expression coming out and will limit the impact of your antibody potentially. So because of all this, we built a proprietary technology. HLA-G is actually active in the form of a dimer, and we kind of built through not through an antibody-based binding, but through a protein-based binding, meaning we're using the receptor to be able to identify and tackle the cancer cells. And by doing so, we were literally flipping things above the head by swapping the immunosuppressive effect that these receptor ILT2 and ILT4 are doing, and making them swapped by a NK cell activation, which will make our cells active and start killing. Here I want to emphasize it's been a year now that we have been working on this, but in summer 2023, our team did a large screening of the now over 90 different activation domain to be sure that we can enhance the cells and improve persistency. Here you have a few of the candidates that we have selected and identified that led us to an oral at ASH last year. The 2 impressive things is not only some of our activation domain are significantly impacting the killing and enhancing our NK cells in killing cancer cells. This is an in vitro stress test, to a degree of 1:40 ET ratio, 1 engineered cell, 40 leukemia cells, and we eradicate them almost in many cases. Actually, today we have 1:80 data that are showing that our cells are literally blasting leukemia cells. You can see the illustration on the bottom. When you have orange, it's our engineered cell. When you have green, it's cancer cells, and you can see that, for example, MyD88 DAP10 or MyD88 DAP12 Zeta are really strong activation domain included in our construct. On the right side, it's about the growth of our cells. Our cells are growing pretty well, which is allowing us to really also have a good expansion and a lot of cells able to chase cancer cells. We replicated this data that were presented at the AACR and ASGCT in solid tumor. Here you have 2 models in bladder and in pancreatic. The model in pancreatic is considered one of the hardest to kill. And like my CSO likes to say, if we can kill this, we can probably kill everything. And we have replicated, this is a 1:10 ET ratio, which would be already a very good results, but we have more data coming in. And on the bottom right, you have the expression of HLA-G that is confirmed that there is high expression in those tumor cells. Based on this, we are moving forward with a seed round that we're actually in the middle of it. I'm actually currently in due diligence with a lead investor and spending my time between speaking and talking to the investor and negotiating our terms. Hopefully, we'll be able to close this lead investor, and we have a goal of raising 5 million by end of the year to support the early development, moving to a proof of concept in vivo. We have preliminary data to be presented at ASH with in vivo in AML, but we want to do more. We also have a third option now, a third large hematologic malignancy that we keep for now undisclosed, but that we will be able to move forward. And we will prepare a Series A next year. So success looks like being able to, of course, close this seed round, launch the Series A. What changed since I did that slide? So we still try to get an interact meeting with FDA early next year. And the idea will be to have not the development of 2 IND packages, but 3, one in AML, another heme, and a basket approach in solid tumor that will be HLA-G positive, with a goal of a first in human around 2027. So I'm going to stop here and be welcoming any question here or later on if you want to talk to me. Thank you very much for your attention.